I'm going to tell you how cryo-electron microscopy is made it possible to see some of these dynamical changes.
First of all, I have to start from the bottom up because I don't know your background. We have to appreciate, first of all, the universality of life forces on Earth. All these organisms that I display here really function on the same principles, and they are all made from building blocks, and they are using very similar metabolic processes. And it has become even more clear as we find out more about the molecular processes. Here is sort of a schematic diagram of the animal cell, where you see there's quite a lot of compartmentation in two different sections. So the cell is not a universe all by itself. Here's a bacterial cell, as depicted by somebody who has done these amazing drawings. His name is Godsell. And you can see that the cell is actually very crowded, and that there are many different structures in there. And in fact, he also depicts ribosomes in there, lined up on the mRNA. And just to give you an impression of the various components. And one of the main components are the proteins, and then a very important component is RNA and DNA, and both are called nucleic acids. And then another major component is lipids. So these three components, proteins, nucleic acid, and lipids, are sort of the main building blocks of the cell. Now, first of all, before going into the real substance, I have to impress you with the difference between a cartoon and the real thing. So this is more closely to the real thing, and this is a cartoon version. You can see that it just depicts the outline. We sort of see that this is referring to the same thing. And in the same way, we are making use of cartoons and very abstract renditions when we go into the molecular realm. So, of course, we never can see molecules in real life. We have to use very powerful instruments such as X-ray crystallography or electron microscopy. And then we wind up with these kind of depictions. And this here is the so-called van der Waals surface, which is, for all practical purposes, the sort of the outer surface of a molecule. Here's a ball and stick representation, where essentially each atom is represented with its bonds. And this here is a ribbons diagram, where only the outline of the skeleton is shown. So when I go into the lecture, I'm going to make use of all kinds of representation. They always refer to the same thing. They're simply different abstractions. So to understand protein synthesis by the ribosome, one has to understand first how proteins are made. Well, proteins are made from 20 amino acids that are strung together in a linear fashion. And this lineup of amino acids is called a polypeptide. And the polypeptide then folds into a secondary, tertiary, and finally quaternary protein structure. So it folds in a very unique way, which is determined by the lineup of amino acids. And only in this folded conformation does it have a real function, because then it has a particular surface properties and grooves and so forth, which lock in with other molecules and make the molecular interactions possible. Now we come to the makeup of RNA. These are sugar phosphate backbone and 4 bases. The 4 bases are lined up there in a particular way. And the four bases are adenine, guanine, cytosine, and uracil. So this is RNA, and you can already see that there's a certain lock and key relationship between bases. Particular bases recognize each other, and they are form just the right distance. So the primary structure here produces a secondary structure arrangements which are characterized by these kinds of lineups, which then lead to helical structures, very well defined helical structures. So the proteins interact with one another and with RNA and DNA. And these interactions make up the metabolism in the cell. The example of a metabolic product, for instance, fatty acids are made by fatty acid synthetase. So the fatty acid synthetase is a very large molecule, just a little factory, which is only making fatty acids. Another example, ATP, which is a unit of energy currency along with GDP, is made by a molecule which is called ATPase, which is a beautiful little structure which is sitting in the membrane and actually rotates as it makes ATP. And other proteins are busy as channels or as part of gigantic molecular machines, such as the ribosome. Here's an example for the job of proteins. Here is one that reads DNA. It recognizes DNA in the sequence of DNA. Here I make use of a ribbon diagram for showing the protein. And as you know from before, this is not the real way the protein looks. We have to flesh it out, and then we see it is more like a substantial surface structure. The proteins in the cell all interact with one another, and they have been, most recently, there have been studies by mass spectroscopy, which indicate the various interactions of molecules in the cell. And then one can make an interaction diagram like this that show the extent of mutual interactions among proteins. And it turns out that there are so many interactions found, most of these are completely unknown. We don't know what the nature of these interactions is. And only a very small part of these is known so far. Now, we have to appreciate the fact that each cell has the information to make a full complement of proteins. And which proteins it makes at any given time is a function of the cell's specialization. So there is, as the cell develops, it goes through a process which is called commitment, and then it can only do a particular subset of its proteins. So examples for specialization are nerve cells, liver cells, skin cells. They only express a particular variety of their protein, even though they have the entire set of genetic material there. Now here we come to a fundamental problem of cell division and inheritance. How can we make copies of something that is three-dimensional? How do we go from here to here? Obviously, we need this kind of duplication or replication. We all know Xerox copy that goes from two-dimensional to two dimensions. That seems a very, very easy process because it involves some kind of contact.